Thank you, sir. I'd like to thank the organizers primarily for keeping our talks early in the morning, because and there's a lot of stuff that you have to carry home. You're used to statins and you're used to bisphosphonates, but what Suja has talked and what I'm going to give you now is a little look into the future, and I hope some of it you can retain, because all of this is not fiction. This is going to become reality in the next few years. Actually, when they told me targets earlier, I was just wondering whether, like we have the lipid targets and the diabetes targets, are we looking at new targets in osteoporosis management also? So I did find one or two references to that effect. JCM 2013 actually carries an article and targets in osteoporosis. And so they're looking at BMD changes, they're looking at bone marker changes. But once I got the whole topic in my mind, I realized they're looking at sites where the drugs can work. So I'm going to quickly spend most of my time on the second and the third part, where we look at the biology of the bone, what are the different cells, how they talk to each other, and what are the novel targets that we have. And just maybe a touch on the new drugs that are available or coming up. So osteoporosis is a huge problem, and to put it in simple words, it's a deficient or quantity of bone, but with loss of quality of bone. And the bottom line is that the patient is prone to fractures. So the fact that the bone has gone down doesn't matter, because we've got a lot of 80, 90-year-old walking around very fine. But the fact that the risk for fracture is high is what troubles us. And the whole effort is to prevent these fractures. And it's a part of aging, so you can't deny that bone mass is going to go down. But when you say that 20% of women are at the risk of getting a hip fracture, and that the important part is that once you've had a fracture, your risk of a second fracture in the first year is pretty high. These are things which trouble you and make you take notice of this disease. Otherwise, osteoporosis is always called as a silent disease. And you have a peak of fractures here, which are generally traumatic fractures in the early second, third decades. But if you look at these osteoporotic or fragility fractures, you have the Colles fracture, which starts increasing about 50, and then you have the hip and the vertebral fracture, which goes up after menopause. So this is what we're trying to prevent. The numbers are huge. I will just tell you that the morbidity and mortality associated with the hip fracture is more than your ischemic heart disease and breast cancer put together. So just to give you an idea, a perspective to our physicians, that we are dealing with huge numbers. And one hip fracture, as the Canadian Orthopedic Association says, is more like a death sentence. It's not a good orthopedic surgeon fixing it up. If you see 20% death in the first year, 36% death in men, large number of them end up being dependent, so either in nursing homes, unable to walk independently, unable to carry out their day-to-day activities independently. So it's a huge problem. The vertebral fractures may not be that bad, but still the numbers are pretty big. So it does end up with morbidity and some increase in mortality. So what are we talking in terms of bone? See, bone is not one piece of tissue which stays static. There's actually turnover happening every day. And so you have the basic multicellular units which are formed of bone resorption and bone formation cells, the osteoclasts and the osteoblast. So up till now the focus has predominantly been on these two cells, but we should not forget the third cell, which is a quiescent cell actually, the osteocyte, and which seems to be becoming a very interesting focus of a lot of experiments, but as of today I'm not going to touch on that. Most of the activity actually takes place in the trabecular bone and less in the cortical bone. So to put it very simply, you have that's the bone surface, and you've got these osteoclasts which keep chewing up the bone, and behind them is osteoblast. So this is a very simplistic view of what is happening in bone cells, and this goes on throughout life, very active during the growing phase, but it slows down, and once you're in the 50 plus postmenopausal stage, the bone formation tends to lag behind, and that's why the sum total is that you start losing bone mass. And in this whole process, which is what my talk is going to be focusing on. You've got factors which control bone the resorption. You've got factors like WNT, the LRP5/6, and the beta-catenin pathway. And then you've got connections, interestingly now, from the brain, from the gut. We keep talking of gut as an endocrine organ. So now gut also controls the bone metabolism. And as I said, any imbalances in this is going to lead to osteoporosis. And so you've got multiple targets now which nature has given you. So what started at the bench now is coming to the bedside. So if you were to quickly look at what is happening in a normal bone, so this is a resting quiescent bone, and then the osteoclasts start working on the bone surface, very much as what happens on our roads. Someone first comes and digs it up, and then another group of people who come and make it. So the osteoclasts tend to eat up probably what is defective, and then the osteoblasts come and sort of take care of it, and that's a very simple thing that goes on day in and day out. But there are many factors regulating this, and I talked about RANK-Ligand, which I'll be alluding to more and more as the lecture goes on. But you've got other prostaglandins, TNF-alpha, interleukins, which also increase the activity of osteoclasts. And on the other hand, because you can't just have bone resorption, the bone formation agents which control the osteoblastic activity are also there. And so you've got OPG, which actually controls the RANK-Ligand, and estrogen, TGF-beta, that's the bone morphogenetic protein. So these are also factors which control the osteoblastic activity and increase bone formation. And why is this necessary really? Because you keep getting defects, you keep getting microfractures day in and day out, and these have to be repaired. Because if these are not attended to, over the years, they're going to become major problems and fractures. So I've already given you an introduction to the various names that you're going to hear through the lecture, but I think probably in this whole show, the most important cell is the osteoclast, because osteoclast is the one which gets things moving. And the important thing is it starts from the multipotent progenitor cell, gets converted into promonocyte, and which then goes into the blood stream, or it stays in the bone with multiple genetic factors working on it to become a pre-osteoclast, and which then gets sort of activated into a mature osteoclast by the stimulation by this ligand. So that's the receptor activator for the nuclear kappa factor B. So we'll still call it just as RANK. RANK is the receptor, and the ligand stimulates the osteoclast. So we'll see where the RANK-Ligand comes from. So as I mentioned, its main function is to resorb, and it's got a highly specialized structure. It's got a ruffled border at the bottom, which actually eats up the bone, and it produces a lot of enzymes. It's got carbonic anhydrase, which is active in producing H ions. It produces tartrate-resistant acid phosphatase. It produces cathepsin K, which is another very interesting target, and integrins. So if you were to inhibit these enzymes, you would inhibit osteoclastic activity or bone resorption. So if you can inhibit RANK-Ligand, you'll reduce osteoclast maturation. If you can inhibit these enzymes, you'll inhibit the activity. So we already have some targets that we can look at now and in the future. So this is the whole thing, how you can control osteoclasts by reducing the formation, reducing number, reducing activity, or reducing the lifespan. Now, on the other side, you've got the osteoblasts, which are the bone-forming agents, which I told you, but they produce these pink little factors called OPG, Osteoprotegerin, and they work as a decoy for binding the, so these are the RANK-Ligands actually, which will go and work on the receptor to stimulate the single nucleate osteoclast into multinucleate osteoclast, and then the activated osteoclast. So the bone formation actually stimulates the bone resorption, and later on we'll see that the bone resorption actually then brings about more bone formation. So that's how nature has kept it. But if this was an ongoing process, there's going to be a lot of destruction going on. So at the same time, nature has also produced another sort of substrate which allows enough ligand to go to the receptor, and then at the same time the OPG which the osteoblast produces would take care of some of these. You have another diagram for that? Yeah. So the OPG which is produced takes care of the extra ligand that is being produced. So we don't want too much of resorption also taking place. So that's how the bone formation cell, the osteoblast, stimulates osteoclast, and at the same time when it's extra, it also controls it. So RANK-Ligand stimulates the osteoclast maturation, OPG controls RANK-Ligand. So the interesting part obviously starts from animals. So we've got mouse studies. When you have the RANK or the RANK-Ligand knockouts, the bone formation increases, and if you have an OPG knockout, you're going to have increased bone resorption. And so you've got osteoporosis, fractures, things like that. And the same thing is also seen in human disease models, like familial Paget diseases, and there is an OPG, autosomal recessive inactivating mutation. You've got juvenile Paget's disease. The other side of the story, bone formation. So you've got a couple of things on the osteoblast, which in the quiescent stage are not active, but you've got this is something which is very interesting. That's the LRP5/6 receptor, which is also controlled by serotonin. I talked about the gut and the bone. So the serotonin seems to work here, and this is a fizzled receptor again on the bone surface. But in the quiescent stage, you have the GSK3 protein, which sort of inhibits what is happening below it. So it's a downstream inhibition. And if you were to work backwards, you need the nucleus, you need transcription to start here, and the beta-catenin is the one which comes to the nucleus and stimulates the bone formation, RNA, etc. So what happens is, when there is stimulation, the main player is Wnt. Wnt brings the fizzled protein and the LRP receptor together, which then downstream, it's again a same GMP, G protein sort of thing, which downstreams work on inhibiting the inhibition, so releases the inhibited beta-catenin to go into the nucleus and bring about transcription of the bone formation units. That's why I said it's good we are early in the morning so that we can sort of still catch on what is happening. So to again look at what is happening. So this is the activated state of the osteoblast. The Wnt, the LRP5/6 and the frizzle protein all together, removing the inhibition of GSK3, allowing the beta-catenin to go into the nucleus and bring about messenger RNA synthesis and downstream protein formation. But at the same time, nature always keeps a control. You just can't have bone resorption all the time or bone formation all the time. So you've got two controlling factors, the DKK receptor and the sclerostin. So these two inhibit the Wnt. They inhibit Wnt signaling. So in a very simplistic thing, this side is bone formation, and on this side, nature keeps the Wnt under control. So you've got targets, either and sclerostin or the DKK receptor or something to stimulate. So you inhibit this side, you'll have more bone formation. You stimulate this side, you'll have more bone formation. So that's the summary of these last three slides. If the Wnt is active, the LRP and the FRZ come together, stimulate beta-catenin, bone morphogenetic protein 2 synthesis, and that's how the osteoblast works more. And at the same time, if you look at what are the defects from mouse models, so if you have a loss of function in LRP5, you're going to have less bone formation, and similarly, the Wnt LRP5/6 or the beta-catenin pathway is essential. So any defect in this is going to lead to bone loss. And again, I said serotonin has become another important thing in experiments, because they find that the gut-produced serotonin actually inhibits or modulates the bone formation. So that's another organ or another target that you have for bone modulation. Now what happens when you come in terms of targets we're talking about is, I said if you do not have sclerostin antibody, so there is plenty of sclerostin, you're going to have inhibition at each of these sites. That's the maturation from the mesenchymal stem cell to the osteoprogenitor cell to the pre-osteoblast to the osteoblast. And if you're going to have antibodies against sclerostin, then you're going to have more maturation and more bone formation. So it's, if you look at it, it sounds very simple. We talked about the osteoclast being inhibited. We are talking of the bone formation cell being inhibited or being stimulated. And actually the answers came out from these studies on the knockout mouse, which are sclerostin negative. And they found that they had very much increased BMDs and the lumbar spine and the femur, more than 50% increase. And similarly, if you had antibodies against sclerostin treatment of overectomized rats at the end of 6 months, they had complete reversal of the effects of 1 year of estrogen deficiency. That is the bone loss that happens with estrogen deficiency, and the bone mass increased not only to the non-overectomized state but even more than that. So these are just some photomicrographs to tell you how the bone mass can actually increase to even more than normal when you're using these antibodies against sclerostin. So with these targets in mind, the last part of my talk, I'll just tell you quickly what are the drugs that are there on the horizon. So we have got bone resorption inhibitors, which are the RANK-Ligand OPG pathway drugs, and then you've got Wnt beta-catenin pathway stimulators, which are basically sclerostin antibodies. There's a whole list of other drugs which are going to come up, but I think what we need to look up is at sclerostin inhibitors. RANK-Ligand Denosumab is already in the market, and Odanacatib, I'll just give you a few lines because looks to be a wonderful drug but has run into trouble. So basically these are the two which are there, promising and very near on the horizon. Denosumab has been around for a couple of years, and now it's also available in India for the oncologist. But I think in another few months, even endocrinologists and physicians will have access to Denosumab, which is a decoy OPG, which reduces the presence of RANK-Ligand, and because of less RANK-Ligand, less activation of osteoclasts. Okay, so that's the thing being repeated. Interestingly, it's a 60 milligram dose given subcutaneously once in 6 months. Side effects are rare, but still you have the serious ONJ and atypical fractures of femurs which have been reported with Denosumab. And the multiple freedom studies have shown that the increase in BMD is fantastic. It's more than double, 68% decrease in vertebral fractures. Actually, the bone mass increase here is seen than what is seen even with teriparatide. It's more. The second class of drugs, Odanacatib, looked very promising. 50 milligram oral once a week. It inhibits cathepsin K enzyme. So that's the enzyme which actually goes and eats bones. And long-term studies showed very promising results. Fracture reduction at the vertebra, not so much at the hip, but still good. But the bone mass increased 11.5% versus 0.3% at placebo, which is more than what was seen with teriparatide with Denosumab also. The major problem is skin reactions, and now the US FDA has actually decided not to allow it to come into the market because of an increased risk in strokes. So Odanacatib would not come in, but there are more drugs in the family, and just like you saw with the CETP inhibitors, the first, second, third failed, the fourth succeeded. Maybe you will hear more of the cathepsin K inhibitors in future. We've been using teriparatide for a long time in the anabolic agents, and we've been looking for more anabolic agents. So the teriparatide actually increases the bone formation through increase in IGF-1 and also inhibiting sclerostin expression. But what is exciting is Romosozumab, which is an anti-sclerostin antibody. And I've already told you how sclerostin reduces the osteoblast function. So if you have anti-sclerostin, you're going to have more bone formation. So that's where the anti-sclerotin antibody is going to work. So downstream everything is going to increase. You're going to have more bone formation. So Romosozumab and Blosozumab, these are the two prototypes, and these are given once a month or once in 3 months. Very significant increase in bone BMD, 11%, 14.9%, 8%. So these are very impressive figures and hold a lot of promise. Drug trials with looking at the dosing has also been, they have been going on. A lot of phase 3 studies have come up, and just one of them here for your information. 2-year study with Romosozumab and then followed by 1 year of Denosumab, and you find significant increase in bone density, which further increases 1 year when you use Denosumab every 6 months for 1 year. But what was disheartening was earlier this month, US FDA again found that there were some problems in terms of higher rate of serious adverse cardiovascular events. So probably they want to be extra safe. They've asked for more data. So you're not going to see this also for some time. But they hold a lot of promise. But abaloparatide, which is a PTH related peptide, is nearly there in the market, and earlier this year in the Endocrine Society there was a lot of discussion on this molecule. So you're going to have at least one promising drug in terms of bone formation. I'm not going to give you anything more except for this thing that you're going to hear a lot of gut serotonin in bone formation, the DKK1, which is involved in glucocorticoid increase of bone loss. This is also going to be coming up. The BMP protein I just introduced to you as a bone formation protein, IGF-1 and bone. So growth hormone producing local IGF-1 in the bone is going to help bone formation. So to conclude, bone is a very interesting organ. Osteoporosis is a very serious disease. It's a silent disease. We need to work towards first prevention. And so whatever I have talked today comes actually second. We need to talk in terms of prevention, in terms of thinking of osteoporosis, diagnosing it early and working on it aggressively. But we could have a 2-day symposium on when to start treatment for osteoporosis, because if you find low bone mass at 35 and 40, you don't run into treatment. You need to do other non-pharmacological things like diet, exercise, choosing the right drug for the right duration. So we were talking about targets in my first slide. Even how long is also a target. But yes, appropriate therapy does reduce the fracture risk, and it's very interesting what is going to happen in the next 2-3 years. I'm going to leave you with these thoughts. Thank you very much for your patience here.